So thank you for the introduction, and thank you for the opportunity to present our work today. I'm Georgia Drakakaki, and I am at the Department of Plant Sciences at UC Davis. Our lab investigated aspects of pistachio development from roots to fruits to the abscission of fruits, and I'm going to tell you some of these things today. We are going to cover cell and hull split, kernel pigmentation, a follow-up from what Phoebe already started, and our efforts on improving mechanical harvesting. So let me start by talking about cell split. While we all like very much pistachios, we know that these are very unusual fruits. They have a hull outside. Some of the people, sometimes when we talk about it, they don't even realize that it happens. And the shell and the hull grows to almost full size before the kernels start to develop. The kernel is green. And another interesting thing about pistachios is that the cells split without even having a dehiscence zone. So we all would like to have this perfect pistachio that the hull is intact, the cell is split, and not those unsplit nuts that sometimes you might see, or hull that is degraded, and that can cause a lot of issues. So let me start by talking about cell split. And let's start by looking at a section of pistachio, a horizontal section of pistachio. We'll go from outside to inside. Here is the hull. Here you start to see already the cell developing, and here is your kernel. These are the definitions of the different layers that you get. The exocarp and mesocarp make the hull. The endocarp is your shell, and then your seed is your kernel. Where does actually pistachio start to split? It starts to split from the side that's kind of more curved, and it starts actually to split from the apical side. But what's actually very different from pistachios to, let's say, walnuts or beans, but let's compare them with walnuts, is that walnuts have an abscission zone. They have this suture site here that actually is made out of different types of cells. It's like a zipper. You can take that zipper, you can unzip the walnuts, and you can crack them open. Unlike walnuts, pistachios don't have that, and still they crack open. Well, how do they do it? My graduate student, Susan, started asking these questions and took an anatomical approach to it. What she found out, in a very, very simplified summary, is that indeed, at the site where we have the shell split, at that belly side that it starts to crack, actually, this suture is thinner. And she started to compare different type of varieties, and she saw that indeed, if you compare, let's say, Pistachio vera to Pistachia atlantica, that does not split, Pistachia atlantica did not have that natural groove that we would still see in Pistachia vera, and you would be able to open it. So that was the first thing that she noticed, that was a pretty strong difference. In the pistachio nut itself, at the shell, if you go to the side that splits, which is the dorsal side, compared to the side that doesn't split, we don't have that difference. So naturally, pistachio knows where it has to split, and it starts already to program the cell to become thinner so that then some forces will just break it open. The next thing she did, among other things in this study, but I don't have time to talk about all of them, is to look at the type of cells and the size of these cells. And she noticed that the cells at the point of split, they were flatter and they were smaller. And the cells at the sides that they were not at the split part, they were bigger. And so these two together could create a difference in strength, in mechanical strength, that will help the pistachio cell open. So if you want to hear more about that, so she was very happy to see the cover when she published her story, but also we are very grateful to Phoebe because we have a little podcast and you can hear more about the discussion in this story. So I'm going to move forward from cell now to hull. Oh, I forgot to mention about this one. We are now looking into mechanisms that they cause this. So we've taken samples from the different sites of cell, whether this is the site of split and the site of non-split, and we've done gene expression analysis, and we are looking at genes that they actually are involved in this particular process. So I hope to be able to update you in a year's time or so when this study is completed. So moving from cell split, I'm going to tell you more about hull now. We all would like to have that perfect intact hull. But we've all seen that indeed, compared to this intact hull, we can have phenotypes that they are all the way cracked, separated across the hull completely, or those that they kind of tear apart, those that we often call tattered. Sometimes we can also see nuts that they have both tattered and separated across hull, that we also call them cracked. So then what Susan, with a lot of other people in my lab, did was to take different fruits from different varieties and test whether there is a difference between the hull split and the hull degradation between them. And what she actually found is that Golden Hills had a higher rate of tatter hull, Lost Hills had the higher rate of cracked hull, and Kerman was the variety that actually had the lowest rate of both. What we also saw was that when we analyzed samples from year to year, even from the very same tree, that's the identity of a tree, that she was looking at the Gulf Hills, she would see differences. So potentially some other factors that they are not related with management, that they are not related to the variety, can contribute also to the different phenotypes of hull split. So as cell biologists, then we said, what actually hull is made of? Can we take a closer look into it? Can we look at the different cell layers and the different cells that they make up this tissue? And so here you can see a section of hull from top to the point where it starts to connect with the cell and stained with different methods. This is Evans blue, this is autofluorescence, and this is toluidine blue. And this is a schematic representation of what actually Susan found. She found that indeed there are different layers of cells, and those that they are very close to the cuticle and the epidermis, she calls them now hypodermal parenchyma. And then she also saw that there are cells under that layer that they are a little bit different, and she calls those filler parenchyma. But she also noticed is that the hypodermal parenchyma, they were more flatter cells and longer, and those that they were filler parenchyma were kind of bigger and fatter, if you wish. She also noticed that there is a transition zone between those and between the hypodermal parenchyma and the filler parenchyma. So even though hull is one thing that you take, and you take it off from the fruit, it does exist from different layers, and within these different layers, potentially, there is a transition zone between different cells. So then, she formed a hypothesis that what happens with the different phenotypes that we have here, based on what she saw with many, many samples that she analyzed, when we have a cracked sample like this one, you will get a separation all the way through all the layers until you reach the shell. When you get tattered, you probably, within the transition zone between these layers, you will start to get a peel off. Sometimes you will have a combination of the two, so you'll get both peel off and a crack all the way through. The next thing that we did was to take a genetic approach and try to find out genes that they are differentially expressed when the hull is cracking or degraded, or when it's maturing compared to fruits that they were less mature. What Susan found was genes that they were involved in cell wall modification. Many genes that they usually are associated with ripening. And when we found that, for example, we found genes that they were involved in pectin modification. So she went ahead and looked into pectin on hull as it was maturing. And she saw that indeed, during maturation of the hull, in this particular case, you will get changes in pectin composition. We use here antibody staining on hull section. Here is a different antibody. And you can see the 90 days after pollination and 130 days after pollination, the difference between the two. So we knew that cell wall was important, and we knew that pectin modification from the cell wall itself was also important in order to allow that separation of cells, and so the hull can break. The next thing we had in also looking at samples that we were getting from different plots that Phoebe was working on is to see the hull split in different irrigation trials. And indeed, we saw that with higher irrigation, we had higher percentage of either tattered or cracked, or in other words, hull split. We also found that for those that they were cracking, they would also go and find that thin section in the hull, and so from there, the separation would happen. So to summarize that, this is the model that Susan developed on hull split, in which you get an intact hull, cell wall is nice, well established, no areas where there is mechanical weakness. And if you start to get cracking, or if you have tattering, you will start to have low cell-to-cell adhesion, cell wall modification, and then you can either have tattering or cracking. So in summary, modification of cell wall is very important for hull split, and the fruit water status also is important. What we also found was a difference within different varieties. And I'll take a minute now and let the audience take a little QR code quiz on answering a question related to hull split, if you don't mind. So if you can take your phone and scan this, you can just answer that question. So I'll leave about half a minute for that. All right. Maybe you don't need the whole minute. It was just one simple question. All right. Let me move now on kernel pigmentation and follow up where Phoebe has started already. The phenotype has already been presented by Phoebe, which is staining of the kernel. And one of the varieties that has the highest phenotypic rate was Golden Hills. So project scientists in my lab, in Min1, really put a lot of effort in this project to identify what is behind that phenotype and characterize it in terms of kernel development too. So by taking samples week after pollination, she noticed that the phenotype only appears when you get completely full cotyledons. This is the stage where you start to see the phenotype. Then she took a biochemical approach to find out what's behind that staining. She took samples from regions that they are not stained and samples from regions that they were stained, that they had higher pigmentation. And she found out that this region was rich in polyphenols. Hence, Phoebe now describing this as polyphenol deposition phenotype. She found high correlation also with different polyphenols. And so then she went ahead and did painstaking job just week by week, taking samples and analyzing for different polyphenols. And you get here the maps for the different polyphenols biochemically analyzed and detected, and seeing what is the trend over 19 weeks. You'll see that at the 19th week, somewhere there, you get several polyphenols that they are increased in the nuts that they have increased color. So she knew that after this careful study, we have about 9 polyphenols that they showed significant enrichment in the samples. And so now we can correlate, and we know that these polyphenols are associated with the kernel pigmentation. Oh. Let's see. Okay. I don't know how to go back. Maybe this? All right. The next thing also, once you get the product of the biochemical reaction, you want to know if the enzymes are also enriched in this region. And again, this slide here shows you that one of the critical enzymes in this pathway, PAL, was also enriched in the colored region. So we do have biochemical evidence not only from the product, but also from the enzyme for the polyphenol deposition. So this summarizes pretty much the study, in which we have week by week analysis of different polyphenols. We know when the phenotype starts, and that is in this region we have enriched polyphenol accumulation. We like to confirm our data by different methods. And in this case, I was very lucky to have a collaboration with Berkeley National Lab, in which they have an imaging approach that you can actually take a section and map chemically a section of kernel. And if you look at the IKD region, the region where you have the color, actually indeed, we noticed that we had peaks of phenolics that was related with the biochemical analysis that we have done. So now, coming from different sources of evidence, we can conclude that the phenotype that we are looking is associated with polyphenol deposition. The next thing that we also wanted to test was whether insect, heat, or pollen are associated, and already you heard some of that from Phoebe, with insect and fungi with the caging experiments. That was pretty much excluded. Similarly with temperature, some experiments that we set up with the help of Themis. And then in pollen, we did not test a lot of different genotypes for pollinizers, but the ones that we tried did not have a statistically significant difference. Again, this is a limited study, so potentially with different pollinizers that Julia includes in her study, there might be a more detailed analysis and more information to get into the effect of pollen. And as already Phoebe described, over-irrigation was co-occurring with the phenotype, where actually deficit irrigation in collaboration with Julia's lab was actually associated with lower rates of the phenotypes, although this was not statistically significant in our data at this point. So to summarize, just a kind of 2 key points from this study: we can conclude that the phenotype of the color pigmentation, it is caused by polyphenols. These are antioxidants anyway. And there is an effect of the of the environment. In this particular case, it was demonstrated with increased irrigation in the particular study. So I do have a few more minutes, and I want to move a little bit into a different area that we started also to explore the last few years, which is to improve mechanical harvesting. And of course, this motivation comes from the fact that we like to reduce the number of sakes that we can have, whether Kerman is to reduce it significantly. Sometimes you might even need many sakes. And so then, towards this end, we started to look into the fruit removal force that is required to be able to take the fruits out from the tree, and therefore separating the nut from the pedicel. And so, Mirmin again, the project scientist in my lab, took samples from different weeks, looking at the fruit removal force, and so that if you get to a point where you can reduce the fruit removal force to approximately 200 G, that's a very nice threshold that you could potentially knock about 80% of the fruits. With that in mind, then, we thought, okay, how about looking into fruits that we can get them to that point and supplement them with a chemical agent. But before going to that, I want to say that at that point of the transition, when you are reaching at the 200 G, which is now we call the mature fruits compared to the immature in terms of abscission zone development, you see significant changes in the cell wall at the abscission zone. Callus is here before, a lot of it. This is degraded when the abscission zone is maturing, and the pH in the abscission zone in this area also starts to change. So we can see actually that the abscission zone is maturing. So then the next thing was, can we supplement it with growth compounds that potentially can affect the abscission zone formation? And we used an ACC, which is a precursor of ethylene. The commercial name for the compound that we used is called acid. The first experiment that we did, it was a dipping experiment. We used 2 concentrations, and we saw that we could actually reduce significantly the fruit removal force. And that projected an improvement in harvesting efficiency. And so then, the second year, which is the year that we just passed, we did have a spray experiment in the field. We had mechanical harvesting, and we used different concentration, 50 and 250 ppm of acid. The variety that we used was Kerman, and based on the data that we have, it looks like that we can have a significant improvement of the mechanical harvesting using the concentration of 250 ppm. So we hope that we will be able to continue this study and accumulate data in the field with spray for mechanical harvesting. So definitely acid is a promising compound, and it looks like it's going to help us improve mechanical harvesting and maybe reduce the shakes potentially from 2 to 1 if necessary. We know that at its effect that it does is not toxic, it's not phytotoxic, and there was no difference in the blanks based on the analysis that we have done, and it does have a significant effect in the reduction of the fruit removal force, So with that, I'm just going to bring all the different things that I told you into a summary, where actually at a cell split, we learned that the shape of suture and the shape and size of cell matters for cell split. For whole split, we do know that the cell wall is important, the integrity of the cell wall is important, the fruit water status is important, and different varieties have different rates of hull split. The kernel pigmentation is associated with polyphenols. These are antioxidants. And there is an effect of the environment. In this case, it was demonstrated with excess of water, with an over-irrigation. With mechanical harvesting, we can see that ACC in the form of acid can improve mechanical harvesting significant. And this is a study that we will continue to follow up to see how we can bring it to a more conclusive area where we can find out concentration that we can actually implement. None of this would happen if I didn't have these wonderful people to do this work. Susan was the person that did all the work, or most of the work, in the cell and hull split, and she had an army of undergraduate students that helped her with that. Unfortunately, she's now moved to Stanford, and I miss her terribly. Min Min developed all the assays for detecting biochemically the discoloration in the kernel, and she's also leading the project on mechanical harvesting. I have been fortunate to have wonderful collaboration, Phoebe, to bring up the project with the discoloration as well, the different trials that she's using, and we are taking samples from here constantly for all this analysis. Julia, Barbara, Gray, Louise, Patrick, and also the folks at the national labs that they help us use their fancy equipment and try Pistachio, and they have never tried that before. The work is supported by the California Pistachio Board. We have support from ANR and USDA and NIFA, as well as our department. So thank you for your attention, and potentially might be able to answer one question.
I did just want to mention that pistachio is not on the seed label, so yeah, just wanted to mention that.
Yeah, so that's why we are looking into several gears of this study, so potentially we could develop the label for that.
So I had a question on the ACC application. What is the timing of that? And then also, besides Kerman, would you be also looking at, let's say, Golden Hills or Lost Hills?
Yeah, so we did start with Kerman because this was the cultivar that we saw that had the highest need for it. That's why we started this study. And for us, in order to do that efficiently, we started to look into the fruit removal force. And so by getting to the point where the fruit removal force was 200, then we knew we can go ahead and spray. We are doing now this analysis with days after pollination and also heat units, and we will be able to bring a model for that so that we can associate all these different elements together into a map where somebody can use it. But I would say that the fruit removal force is one of the better units to go about and spray.
And the reason I mentioned Golden Hills is because with Kerman, if you still, end up leaving some for a secondary shake, you really, at the end, on a commercial basis, the fact that you have to do another shake, we're still not achieving anything. But on Golden Hills or Lost Hills, where we get already more removal, the idea would be to get it so that we can get it all with more removal.
Yes, I definitely see the point, and there might be even more advantages, like trying earlier, you can avoid a lot of unwanted phenotypes, and these are things to explore. We could just do so much for what we wanted, but definitely we can look into other varieties. Thank you.